Good morning, children. Today we are going to discuss about the Mendel's law. So, Mendel's, you know very well, principle of dominance. One gene will be dominant factor, and another one will be recessive gene or recessive factor. Like that, we discussed in the previous classes about the Mendel's law. That is called principle of dominance. We discussed. Next law. The Mendel's second law is principle of segregation. Segregation means separation. Separation of genes. That's it. Very simple. For example, the two unit factors of a character, capital T, small t, character is tall. Two unit factor of character which remains together in an individual do not get mixed up and keep their distinct identity. Capital T, small t, they will not mix each other. They will maintain, keep their distinct identity. This is only called separate or segregate during gamete formation. So each gamete receives only one factor for each character and is always pure, either capital T or small t, that's it. But capital T, small t cannot mix. That is only called principle of segregation. So each character and is always pure. So that's why this law is also called principle of segregation, is also called principle of purity. For example, capital T, small t. Capital T, small t separate, tall and dwarf. They separate during gamete formation. The equal factors, equal frequency in the male and female. That's it. There is equal frequency in the male and female. So this is called principle of segregation. So capital T, small t. That's it. Capital T, small t will not mix each other. Even though capital T is dominant and small t recessive, but they will not mix each other. They will maintain their purity. Only at the time of gamete formation they will be separate. That is why that law is called principle of segregation, or what is called principle of purity. So capital T, small t, capital T separate, small t separate, equal during separate during gamete formation. So the equal factor, so equal frequency in the male and females, that is only called what is called female gametes. That is only called what is called principle of segregation. Two unit factors of a character which remains together in an individual do not get mixed up and keep their distinct identity. Separate or segregate during gamete formation. Each gamete receives only one factor for each character and is always pure. Okay, always pure. So that is important. Principle of purity. Capital T, small t. See, capital T, small t. They are not mixing. At the time of gamete formation, capital separate, small t separate. Again, they will enter into the what you called in the next generation. Capital T, small t will be like that only. So this is only called principle of purity. Separate during gamete formation. Equal factors, equal frequency in the female and the male gametes will be there. That is called principle of segregation. Next one, principle of independent assortment. What is called? The third law is, first one is law of dominance. The second one is principle of segregation. The third one is principle of independent assortment. Principle of independent assortment. So there will be a unit factor of each character is assorted or distributed. So this principle states that unit factor of each character is assorted or distributed into the gametes independent of the unit factors of only other character, and what you call gets randomly rearranged in the offspring. Dervish, you know dihybrid cross, capital R, capital Y, round and yellow, small r, small y, wrinkle and green. Dervish, either that will joins with the capital R or small r, capital R, like that. The table you know very well. So what happened? Gametes independent of the unit factors gene any other character and gets randomly rearranged in the offspring. Randomly rearranged. Their wish. Mendel's dihybrid cross offsprings of F1 generation after self-breeding produced 4 types of offsprings. 2 types were similar to the parents, while the remaining 2 types had combination of traits. This became possible because of the unit factors of 2 characters assorted independent to each other. So capital R, small, what do you call it? When the table is there or not, you know very well the dihybrid cross is there or not. In that dihybrid cross, what you identified, there will be yellow round. Okay, winkle is there or not? Capital R, capital Y, small r, capital Y, capital R, small y, capital small r, small y. Okay, like that 2 are equal to parents, but remaining 2 are what you called. So 2 types had combination traits. This become possible because the unit factors of the 2 characters assorted independent to each other. So independently they attach each other. So the best example of principle of independent assortment is, you can remember the dihybrid cross. In the F1 generation, what happened? Capital 2 parents will come, but remaining 2 pure will come. Remaining, they will be, okay, 2 types combination of traits, combination either small r capital Y and capital R small y, like that the combinations will be there, so they will be randomly will be formed. So the principle of independent assortment, best example is the Mendelian dihybrid cross. Mendelian dihybrid cross. Next one, inheritance of the traits. Mendel proposed that which are responsible for the characters. So, which are responsible for the characters? The genes are responsible for the characters. After knowing that characters are inherited from the parents, the question arises that which entity controls inheritance? This is important. Which entity controls the inheritance? So, characters are spreading from parents to offsprings. Which one? So, Mendel proposed that a pair of factors which are now called genes control the inheritance. The genes are controls the inheritance. Genes are means the segment of DNA. Okay, adenine, thymine, guanine, cytosine, like that is there or not? Togetherly will be form gene. The gene have some thousands of nucleotide base pairs. So the genes only control in the inheritance. So that is the important. Next, what is this one? The parents, both at the time of the gametes and the combination, both male and female gametes will give equal DNA to the progeny during sexual reproduction. So, male give of DNA and female give of DNA for the progeny. So, that is the very important. What I am telling is, according to the Mendelian experiment, both parents must be contributing equal, what is called equal DNA. Both parents are contributing how much? Equal DNA. This is the important. Both parents must be contributing copy of the same gene. Okay, they are transporting the what is called equal DNA. Next, each progeny must have 2 sets of all genes, one inherited from each parent. And for the mechanism to work, a gamete must have only one gene set. You know, gametes must be haploid condition. Male is a haploid and father is a haploid. Female and male togetherly, what you call haploid togetherly form what? Meiosis. Then what will be zygote mixing will be takes place. Then zygote meiosis after haploid gametes were forming. Those haploid gametes are, so what happened? The gametes togetherly form, what you called? The zygote will be diploid, zygote will be formed. So here what you understand is where the DNA is present. The DNA is present where these genes are present. Gene is present, the DNA. So DNA where it is located. So every germ cell will take the one chromosome for each chromosome pair, and this may be either maternal or parental. So every germ cell will take the one chromosome. One is from father, one is from mother, will take and forms the diploid condition. So 2 germ cells combines, they will restore the what you called number of chromosomes in the progeny. So what you called haploid, to haploid will be converted to which one? A diploid condition will be formed. Diploid means two sets of conditions. But gametes always, I even said gametes how much? What is the condition? Haploid. So the gametes haploid condition will be there. A section of DNA carries information for a particular type of protein. See, DNA helpful for what? Protein. DNA helpful replication. DNA to transcription. mRNA to protein translation. It's a character will become. So these all are the proteins are nothing but with here characters. So the genes are responsible for the, so genes controls the characters of the organism. So parent male will give off nuclear genetic material, female will give. So haploid togetherly diploid will give what is called protein gene you know very well. Character will be controls the inheritance of organism. So like this, inheritance will be there of the traits. How inheritance? First you have to know gene. Gene is a segment of DNA. So Mendel proposed a pair of factors which are now called genes controlling inheritance. So parents will distribute the equal DNA. So to the progeny during sexual reproduction. So gametes only one set and meiosis will be takes place. Gametes will be formed. So of n plus n 2n. So 2n is the zygote. So equal amount of DNA given by the parents. The DNA will be togetherly, that chromosomes, diploid chromosomes will helpful for the metasis and complete helpful for what you call DNA to replication and then transcription, the protein to character, then the genes will be formed. Like this, what you call genes are responsible for the characters. Genes are responsible for the character. Do you understand or not? What is DNA? Deoxyribose nucleic acid. Where the DNA is present, this is a chromosome, right? This is a chromosome structure. It's a centromere. It's a chromatids. These two chromatids have. Okay. In this chromatid only DNA is present. DNA is a mainly this much of DNA will be fixed in this particular area because of histone proteins. So this DNA will involve in the transcription. mRNA will be formed. So mRNA to protein formation, this protein only responsible for the character. So DNA consists of genes. Genes only characters. So genes are mainly controls the inheritance of the characters. So that is only what I call inheritance of the traits will be there. So like this, haploid, diploid, zygote, meiosis, when the mixing of the what you called this DNA will be takes place in the meiosis, crossing over will be takes place, you know very well. Diploid, zygote, you know what will be takes place, like zygote, tachytin, diplotin, diaconsis is there or not in the meiosis and mitosis. So mitosis after 2n condition cells will be formed, but meiosis after only gametes will be formed. Gametes means what? Nothing but what is called one single n condition. So these are very useful for the what is called these all the things, okay, for the characters, inheritance of the traits. Okay children, so what are the important points to remember in these laws? First one is principle of segregation. So the two unit factors do not mix it up and keep their distinct identity. So capital T, small t, they will not mix each other. There is no mixed medium. Capital T separate like that only. Capital T, what is called small t. That's it. They will not mix. They separate or segregate during gamete form. Gamete formation, capital T separate, small t separate. Each gamete receives one factor. So for each character and is always pure. So it will always be pure. That's why it is called principle of purity. What is called the principle of purity? Why capital T, small t is there? Capital T, small t will not combine. No medium. Only capital T is separate. What is called small t separate. Capital T creates the what is called tallness. Small t darkness. So these are what they will do. So at the time of gamete formation, they will separate. Again, one will be dominant, one will be recessive in the offsprings. That's it. There is no mixing. That is why that law is called, what do you call that law is called? That law is called, what do you call? The law of principle of segregation, are also called principle of purity. Principle of segregation or principle of purity. This one. Principle of independent assortment. Just you remember for this, dihybrid cross. What is the dihybrid cross? Mendel dihybrid cross offsprings of F generation after self-breeding produce 4 types of offsprings. So 2 types were similar to the parents, while the remaining 2 types had combination of traits. This remaining 2 types combination of traits or not. This became possible because the unit factors of 2 characters assorted independent to each other. So they assorted independent. That's it. Their wish, they will joining each other and forming. So you see so many round and wrinkle and what you call different 9:3:3:1 came or not? Why 9:3:3:1 came? So some will be round and yellow, sound round and some round and green, wrinkled and green. Why? Because the independent assortment, that is called independent assortment. So R capital R Y small capital Y, maybe that will joins with the capital small R small y. That is there. That is why that is called independent assortment. So 2 characters assorted independent to each other, no controlling will be there. That is why the principle of independent assortment we can see in the dihybrid cross, Mendel's dihybrid cross experiment. Okay children, so next, inheritance of traits. How the characters are spreading from one generation to another is simple, because of genes. Genes nothing but factors. So parents will give equal DNA to the progeny during sexual reproduction. Male gamete, female gamete togetherly form 1n plus 1n, what do you call? Gamete zygote diploid. That diploid will be developed into embryo fetus, like that. So what the characters are in the male half and female half, both characters we can see in the offspring. So where this character giving nature, where it is present in the genes, where the genes in the chromosomes, especially chromatin, you know very well. So the chromosomes have the chromatin. DNA mainly, consists of genes. These are involved in the transcription process. mRNA to protein will be, this protein is only responsible for the character, but main thing is DNA. From where this DNA is coming? So male and female gamete DNA is off sharing. So characters only, genes only responsible for the characters. Genes means nothing but what? The segment of the, what you call, DNA. See the chromosome structure. See one set of chromosome, only one set is there, in the sense haploid. 2 sets of chromosomes are there like this. So one set from father, one set from mother, haploid plus haploid, diploid. So they will be mixed together. DNA will be mixing each other, and what will happen? The DNA will shows the new variety of characters. So that is why, so genes, because without genes no transcription, no proteins, no characters. So always should remember why the new characters are forming, in the sense because of the genes. So genes only involved in the transcription process, genes only responsible for the what called characters. That is only inheritance of the traits. The factors are genes responsible for the character. During sexual reproduction, both male and female gametes will be mixed and produce the zygote. That zygote only useful for the variety of new characters will be formed in the new progeny. So DNA consists of gene. DNA located in the chromosomes located in the nucleus. So nucleus, 2 sets of chromosomes diploid, 1 set of chromosome haploid, diploid 2n, haploid 1n, meiosis. After haploid cells, gametes will be formed. So like this, the traits, inheritance of traits, mainly because of genes only responsible, or factors only responsible for the inheritance of the characters. So like that we should have to remember. So it's a principle of segregation, principle of independent assortment, that is inheritance of the traits. Genes are responsible for the inheritance of trait. So next class we will discuss about another important concept. What is another important concept? That is sex determination, how the XX chromosome. I said or not? XX means what? XY means what? So that determination, female, male sex determination, we will discuss.